So we're probably not going to get through all of this today, but we'll complete it tomorrow and then continue on with the next lecture. So sometimes that happens where, we're not able to complete an entire lecture in one class. So we'll continue it in the next and then start the next lecture. But today's lecture is an overview of some of the topics we're going to touch on. We'll go into more detail later. The exception is evolution. The only time we're going to talk about evolution is today. And we're not going to talk that much about it, unfortunately. But if you're really interested in evolution, or if you're a biology major, then you will need to take Biology 151 after this class. And then you'll learn all about biology, or all about evolution. So there are several themes in the study of life. So inquiry is important. Evolution, and we will talk a little bit more about this towards the end of the lecture, is a process of change, right, which has transformed all the life on Earth. And biology, anything that is ology means the study of, so biology is the study of life. And biologists and all scientists like to ask questions, right? So we ask many, many questions. And life cannot be summed up in one sentence, right? There are many aspects to life. We generally refer to life forms by what they do, but we're not entirely sure what the exact definition of life should be. And the reason why is we haven't discovered everything that exists on the earth yet. So there could be things yet to be discovered that might change how we define life. But we look at how life forms reproduce, how they grow and develop, how they make energy, how they regulate their internal environment, how they respond to the environment, adaptations they possess. And many forms of life show a remarkable levels of order in terms of their construction and how they're formed. While it's true you will have to memorize a lot of facts, just memorizing, right, those facts may or may not enable you to pass the class. It depends. You have to understand how these facts come together to form concepts and be able to understand those concepts. So there's no getting around the fact you're going to have to memorize stuff, right? So biology has a lot of words, a lot of terms. You got to know what they mean. But you also have to be able to put those words together in order to discuss concepts in biology. So we can organize biology looking at different themes. Of course, the overarching theme is evolution. Everything we know about living organisms makes sense because of evolution. And the organisms, right, that are currently living are modified descendants of common ancestors. We can study life at different levels, right, from molecules to the entire living planet. And the study of life can be divided into many different levels, right, for organizing the information. And now this diagram kind of shows things in the reverse of how I would talk about them. I would start off with atoms, right? So atoms are the smallest units that we're going to discuss, along with the subatomic particles. But we're going to discuss how the atoms create the elements. And the elements are assembled together into molecules. Smaller molecules are assembled into much larger macromolecules. Macromolecules can assemble to form structures like cells and the organelles found within those cells. Cells can associate together to form a tissue. And then tissues can associate together to form organs. Organs can associate to form organ systems. And that leads to a complex multicellular organism. And those complex multicellular organisms live in populations, and they associate in different communities. So you have many different populations existing in a community. And those communities exist in an ecosystem. And there are several different kinds of ecosystems on the Earth. And the entire Earth itself is the biosphere, where we find all the life forms. And those life forms are in water, in soil, they're in the air, they're even in rock, right? They grow in ice. So, the whole Earth is the biosphere, and we find life everywhere in the Earth. Most of that life is actually microscopic, but there is an incredible number of life forms that exist, and probably most of them we have yet to discover. Now, because many topics are complex, and because they're complex, we reduce them into smaller pieces and talk about the individual pieces, which you then put together, right, to form the larger concept. So reductionism is where we like to break things up into smaller pieces that we can then more easily understand and put them together. An example would be the structure of DNA, right? So deoxyribonucleic acids, it's made up of chains of nucleotides. There are two chains which are held together by hydrogen bonds to make a DNA molecule. An even better example is cell respiration, aerobic cell respiration, which is very complicated. And so we'll talk about a lot of the individual pieces, and then you have to put those pieces together to understand how cells make energy. Now, I have highlighted this in red and have underlined it because this is very important. We generalize, right? So that means that in order for the sake of simplicity, we have to generalize many concepts because these concepts are very detailed and they're very complicated, and there are almost always exceptions. There is not anywhere near enough time to talk about all the different exceptions to any given rule, but I expect you to understand the general rule, the general concept. Organisms interact with their environment, and the environment includes both living and non-living factors. So non-living factors like soil, water, rocks, the presence or absence of oxygen, right? So those all determine what kinds of organisms can live in that particular environment. And those organisms interact with the environment. And those organisms can actually cause changes to occur to the environment. For example, a tree. Trees absorb water and minerals from the soil and also carbon dioxide from the air. And they produce oxygen, which is released into the atmosphere. And the roots help hold the soil together, right, to keep the soil from washing away. And when that tree dies and decomposes, it becomes soil. When you look at beavers, right, they make dams on streams and creeks and small rivers. That dam alters the ecosystem, right? Human beings, though, there's no other animal that's better than human beings at altering the environment, right? We're not going to get into global warming in this course, but that's an example of how humans have altered the environment. But we build houses, we make fire, we make heat, we air condition to make things cooler. So we like to adapt the environment to us, or at least we like to think that we can do that. Ecosystems involve the cycling of nutrients and the flow of energy. So nutrients like nitrogen and phosphorus are absorbed from the soil by plants. And so those nutrients get assimilated into plants. And then animals eat the plants. And that's how the animals get those nutrients. And then other animals eat the animals that eat the plants, and so on and so forth. When those plants and animals die and they decompose, all those nutrients go back into the environment where they can be recycled and reassimilated by other living things. When it comes to energy, most ecosystems on the Earth are dependent on the sun, right? So light energy from the sun, photosynthesis. Again, plants use light energy to make food like sugars, right? Carbohydrates. Animals eat those plants. Animals eat the animals that eat the plants, and so on and so forth. But the ultimate source of energy came from the sun. And that is true for most ecosystems, not all of them. Does anybody know of an ecosystem on the Earth that is not dependent on sunlight or photosynthesis at all?
Like really deep in the ocean where the sunlight doesn't reach.
Right, those deep sea thermal vents, they're deep beneath the ocean. Tremendous pressure that would squash us in an instant. It's completely black. No sunlight whatsoever penetrates. So there's no photosynthesis. And yet there are amazing communities, right? These deep sea thermal vents, which were discovered in 1977. And they discovered these giant tube worms, 5 feet long, blind crabs and shrimp and even some fish. There's all kinds of things. Tremendous pressure. Usually the water is very warm and very toxic with toxic sulfides in the water. And so these ecosystems are completely independent of the sun. No photosynthesis. So they rely on chemosynthesis in order to make energy. So most ecosystems do depend on the sun, but not everything. And that was only discovered back in the late 1970s, so not all that long ago. I know it's long for you guys, but not for me. So energy will enter into an ecosystem as sunlight. It will be used in photosynthesis by plants to make food. Animals eat the plants. Animals eat the animals that eat the plants. And that food energy is converted into ATP, right? The kind of energy that is used in cells. And in the process of converting that, chemical food energy into ATP, heat is also produced, and that heat is lost to the environment. So the energy from the sun ultimately ends up being lost as heat. We're going to talk a lot about energy relatively shortly, but work requires energy, so no work can occur without some type of energy. And there are many different forms of energy. Light energy, chemical energy, kinetic energy, thermal energy. So there are different kinds of energy. And these energies are exchanged, right, between organisms and the environments. And so one of the things that we will learn is that energy is neither created nor destroyed. The energy of the universe is constant, which I know sounds strange, but all the energy that's here now was here yesterday and 1,000 years ago and 1,000,000 years ago, and so on and so on. It's just transformed from one type of energy to another. A good example, a light bulb. And we're talking about like older incandescent bulbs. I don't know if you guys are that familiar with incandescent bulbs, but those are the bulbs that are too hot to touch, right? If you were to touch a light bulb, right, the old-fashioned light bulb, it would burn you. So the electrical energy is being converted into light energy and heat energy, right, thermal energy. But it's the same amount of energy that's produced as light and heat as is the electrical energy going into the bulb. So the energy is being transformed. It's not being created, it's not being destroyed, it's transformed from one form to another. So the solar energy from the sun, the light energy, is ultimately transformed into heat energy by animals, by other living organisms. When we look at structure and function, we see they are related to each other. If you look at a leaf, most leaves are thin, right, and flat. That maximizes surface area for exposure to sunlight, which is what chloroplasts need to photosynthesize, right? So there is a very close relationship between structure and function. When we look at birds and their wings are designed to provide the uplift needed for birds to fly. When we look at the bones of most birds, not all of the bones in their body, but many of them are hollow. And they used to think that the bones of birds were lighter to help them fly, but that's not actually correct. I mean, relative to size, bird bones are normally just as heavy as any other bone of the same size. But they are hollow and they contain air spaces. So there are actually air sacs in the bones of many birds, and those air sacs are connected to the lungs of these birds. And don't ask me how, but these air sacs enable birds to take in oxygen both when inhaling and exhaling. And as we're going to learn, when we learn about aerobic cell respiration, oxygen is needed in order to make a lot of ATP. So birds that flying takes a lot of energy. And so having a lot of oxygen helps them to make all the energy they need to fly. But the hollow bones are not lighter. It just helps them get more oxygen so they can make more ATP. Neurons, if you take anatomy and physiology later, you're going to learn all about neurons and how you have, the neuron, the cell body, the long axons, and the dendrites that make all the connections, right, to help you think, right, to help your brain function, right, to help you interpret, the world around you. And then mitochondria, and we are going to revisit this later, but mitochondria are the organelles inside of eukaryotic cells that make ATP. And as we're going to learn, all of the components needed to make the majority of ATP in a cell are embedded in a membrane. And so we see the inner mitochondrial membrane is very highly folded. All that folding increases surface area. And since all the components needed to make ATP are in the membrane, more surface area means more of those energy-making components. And so relative to that tiny volume inside the mitochondria, you can pack a lot of those energy-producing components when you fold the membrane. So structure and function are related. There is a reason why things are the way they are, why they look the way they do. Now, the traditional definition of life states that the smallest unit of life has to be at least one cell. So we consider cells to be the smallest unit of life because a cell is able to perform all the functions necessary for life. All cells are enclosed by a membrane. Certain cells also have a cell wall around them, but all cells have a cell membrane. And they all use DNA, right, to store all the information to create a new organism. Cells can divide, and that's how they can reproduce. That's how a multicellular organism grows, and that's how damaged tissue gets repaired and replaced, because cells can divide. Eukaryotic cells are surrounded by a membrane. Certain cells, like plant cells, also have a cell wall, but they all have membranes. And eukaryotic cells are typically large. Prokaryotic cells are simpler, smaller. They do have a cell membrane, usually also a cell wall, but all those membrane-bound organelles like mitochondria and chloroplasts, the Golgi, the endoplasmic reticulum, those are only found in eukaryotes, not prokaryotes. There's no nucleus in a prokaryotic cell. And that's what the word prokaryote means. It means pre-nucleus, so before there was a nucleus. Bacteria and archaea. Archaea are the other major group of prokaryotes. And there are two domains, one for the bacteria, one for the archaea. And the third domain, eukarya, is for all the eukaryotes, all the plants, animals, fungi, and protists. So here we see our giant eukaryotic cell, and we're going to learn a lot more about cell structure in detail later. When it comes to DNA and the chromosomes, we're going to spend a lot more time on this later, so I'm just going to go through this relatively quickly. But chromosomes contain the DNA, the deoxyribonucleic acid, which contains the information needed to create a new organism. And the information is stored in discrete sections of DNA called genes. And genes are responsible for traits, right, for characteristics, which can be transmitted from parent to offspring. Eukaryotes have long linear chromosomes. Prokaryotes have small circular chromosomes. Most of the DNA of a eukaryotic cell is in the nucleus. However, mitochondria and chloroplasts also have a circular chromosome. So not all the DNA is in the nucleus, but most of it is. And when it comes to regulating the expression of genes, we'll get into that later on and see it's very complicated. Just because you have a particular gene does not mean you're going to express that particular characteristic. It's much more complicated than that. So the genes of the DNA is transcribed to RNA, which is then translated into protein. And we're going to go over transcription and translation in a fair amount of detail later on. And the genome is all the DNA inside of a cell, whether it codes for a protein or not. And a lot of your DNA does not code for proteins. When it comes to studying genes, we have computers and we have a lot of technology to allow us to sequence genes, right? To sequence DNA, right? To sequence RNA, even to sequence proteins, right? The amino acid structure. And just as an example of how things have really changed, the entire human genome required about 13 years to sequence at a cost of about $1 billion. It was completed in 2004, which means it was started around 1990. If they were to do that today, they could sequence the entire genome in just a few days for a few $1,000s. So the technology has increased dramatically, almost exponentially. And so sequencing genes today is easy. If you go on to take microbiology, which I also teach, we talk about how we do this. And literally, you can extract a DNA, amplify it with PCR, which we're going to talk about in this course in lab later on. You take your amplified DNA and you just drop it off to a local lab. And there is a local one here. I forget what its name is, but you just drop it off. They do the sequencing for you and they email you your sequence the next day. And they only charge like $5 to do it. So it is incredibly fast, efficient, and cheap, right, to sequence DNA today. Bioinformatics is using computers to analyze all those sequences. And we do have some labs involving bioinformatics, so we'll get into that later when we reach that point in lab. Now, when it comes to cells regulating a metabolic process or a metabolic pathway, they can do this through positive and negative feedback. So for example, let's say that a cell is making a certain product. And if the cell has made more product than it needs, then there has to be a way to stop making that product, temporarily, right, to allow the cell to use up that product. And then it can start making more products. So that would be an example of negative feedback, where the cell has made more than enough of something. Maybe they made too much of an amino acid and they don't need it. So they temporarily stop making it. So there are mechanisms to help slow down or temporarily stop, right, the synthesis of a product. And that's negative feedback because it's inhibiting the formation of something. Likewise, sometimes the cell needs to make more of a product. Certain environmental conditions may have changed and the cell suddenly has to upregulate and start to make more product, and it would be positive feedback. Some other examples: during childbirth, oxytocin is a hormone which increases the rate of uterine contractions in order to help push out, right, that newborn. When it comes to blood clotting, you cut yourself and you start bleeding, well, that activates a series of reactions in the blood to form a clot. And so that process gets upregulated, right? There's positive feedback in order to make that clot form as quickly as possible so you can stop bleeding. But this is how cells regulate things. So if there's a metabolic pathway making an end product, the end product can feed back to an enzyme early in the pathway and actually temporarily turn that enzyme off. So that would be negative feedback. We'll also learn about how sometimes the end product can actually help to turn off the gene so the enzymes don't get made. And that saves a lot of energy. This is all about energy, right? Cells, I can't afford to be wasteful, so they need to do things when they need to do them, stop doing them when they no longer need to do that in order to save energy. And then positive regulation just upregulates, increases the formation of something. So back to evolution. Theodosius Dobzhansky made the statement that nothing in biology makes sense except in the light of evolution. So when you look at all the forms of life, how their structure is, how they function, then it makes sense, right, when you think of evolution. So evolution is a unifying theory of biology. So far, we've identified less than 2 million species on the Earth. Estimates are there could be as many as 10 million to 100 million different species of life. Most of them are microbial life, right? So they're not easy to see. One of the things that scientists had hoped to do was the all species inventory. I just mentioned it because their goal was to catalog and identify every form of life on the Earth and do it by 2025. It's 4 years from now. Not going to happen, right? It was too complicated. The funding dried up, so they were not able to do that. But it was a nice idea, but they just couldn't do it. When we start to classify things, we use taxonomy. So taxonomy refers to all the different names we give to things. And so the largest taxons, the largest grouping names are the domains. And then we have kingdoms, and then phylum and class, order, family, genus, species. All right, so the domain is the largest grouping. It used to be kingdom, but now it's domain. I'm sorry, what was that? So a domain, remember there's only 3 domains: bacteria, archaea, and eukarya. So eukarya has all the eukaryotes in it, all the plants, animals, fungi, and protists. And then we have kingdom, phylum, class, order, family, genus, and species. So the way that I used to tell students to remember before we had domain was King Philip came over from Great Spain. Okay, so King Philip came over from Great Spain. And now that we have domain, damn King Philip came over from Great Spain. So once again, we used to have just 5 kingdoms. Now we have 3 domains, and then we have other kingdoms. But 2 of the 3 domains are prokaryotic, the bacteria and the archaea. And the archaea are something, you will learn more about them in Biology 151 if you take that. And you'll learn something about them in microbiology if you take that course as well. A fascinating group of microorganisms. They're often referred to as extremophiles. They love extreme environments like very high heat, very acidic environments, very toxic to everything else. But the archaea can survive there. And in fact, they evolved to live there. Right, so bacteria, archaea, which have a lot of similarities to the bacteria, but they have some important differences. And so they were separated into their own group. And then all the eukaryotes, all the protists, like paramecia, euglena, amoeba, fungi, all the nasty mushrooms and things, plants and animals. Now, when it comes to classification, it gets complicated. And so let's just look at a graphic, which I just included to show you how complicated it can be. So when human beings first started to notice things and say, maybe we need to start classifying things, Linnaeus in 1735, he came up with two kingdoms, Vegetabilia, which were the plants, and then Animalia. Over 100 years later, we had three kingdoms, right? The plant kingdom, the animal kingdom, and then Protista was added for everything that wasn't a plant or an animal. And then we had two empires, right? Prokaryota and Eukaryota, but that didn't really last long. But we noticed how that things have changed. In the early 1900s, right, we added Monera, right, which included the bacteria, right, the prokaryotes. And then in 1977, when the archaea were discovered, those deep sea thermal vents, they had to add archaebacteria, which they later said, no, they're not bacteria, we're just going to call them the archaea. But what we can see is that it keeps changing, right? We had three domains back in 1990, and then we had 8 kingdoms proposed, right, by Cavalier Smith. And then he or she decided, no, maybe it's 6 kingdoms instead. And then the latest, we have 7 kingdoms proposed. But the point is that classification is complicated and it's constantly changing. So if you want to be a biologist that loves to classify things, there's a field of interest for you. But classification is constantly changing. So it does get very, very complicated. Now, a striking unity underlies the diversity of life. So DNA is universal. All the forms of life that we know of use DNA as their genetic code. So unity is evident in many features of cell structure. And then I included there is unity in diversity, which from a biological perspective, we're talking about evolution. From a sociological perspective, we're talking about people, right? Different races, ethnicities, religions, all those kinds of things. But this is a biology course. So for us, diversity refers to genetic diversity. And so we'll finish up here with cilia, an example of a structure that is used in both simple organisms like paramecia, a single-cell protozoan. It's also found in complex organisms like humans, like us. We have cilia in our lungs, right? They line the respiratory tract. We also have cilia in the vagina. But when we look at the cilia in us and the cilia in paramecia, they're built the same way. They're constructed the same way. And yet they're used for very different things. Right? The paramecium uses it to swim around, right? To hunt for food, to escape predators. We use cilia to move mucus out of the body. But it's the same structure, the same design plan. So why do you think we would see the same design plan in so many different organisms? What does that suggest? Right, paramecia have them. And we have them. So what does that suggest?
Common ancestry.
Right, common evolutionary ancestors. And so we have to stop here for today.